I was wondering if they were going to tape.
Yes. I don't know how to get it to use my forward-facing camera, but I guess I'll just do that. Or I can come sit down there. Now I can keep track of this.
Oh, excellent.
Why is it backwards?
Because you're... I think it's because you should be filming it in the other device. I'm not...
Alright, let's figure this out.
I don't want to put my face.
One sec, John.
What?
I wouldn't sit here if I were you.
Why not?
I'm trying to figure out how to record this. So fewer people sitting. Oh, there.
That's where I was planning on sitting.
Okie dokie.
I didn't see the computer at first there, but I assumed that there was a space between the two of you for a reason.
Yeah. I don't know why it's backwards. Alright. Not good. Alright, I guess we're just gonna have to watch it.
You might have to end it to change the direction.
What?
You might have to end the stream in order to change the direction.
I'm afraid of them losing the link. So I'm like, I'd rather it watch it backwards. When I practiced it also, it was only through the front-facing camera, not the back one.
So is it actually showing it in the mirror?
I don't know, maybe it's not showing it in here. I can't actually see the screen myself.
So it is truly my pleasure and honor to be introducing Meagan Rubel today, who's going to be defending her thesis entitled Effects of Diet and Parasites on the Gut Microbiome or Microbiota of Diverse Sub-Saharan Africans. So for those of you who don't know Meagan, I want to just give you a little bit of background information. She comes from a really interdisciplinary background. She did her undergraduate degree at Arizona State in anthropology, working with Ann Stone in her molecular genetics laboratory. She managed to obtain a master's degree of public health en route to getting her PhD, which is really impressive. And when she came to my lab, she had sort of a broad interest in understanding human evolutionary history and adaptation in Africa. We focus on populations from Africa. Now, at the time, my lab had never done any microbiome research. So, in fact, very little was actually known at the time about what is the microbiome diversity like in indigenous populations, and particularly from Africa. So, Meagan had the initiative to basically come up with this idea, take it as her own. And I'm really grateful to you, Meagan, for initiating microbiome research in that lab. I'm also really grateful to Rick Bushman and to his group for offering their guidance and sharing their experience. Without you guys, this wouldn't have been possible either. Now, another thing about Meagan, you may or may not know, if I remember correctly, when you first came to my lab, you all brought cupcakes, am I right? I had another opportunity to try her cupcakes at a later date. They're amazing. She's really, really good at that. She's not only an amazing baker, she's an excellent writer, and particularly an excellent grant writer. So she successfully got funding from the Wintergreen Foundation, the Leahy Foundation, the NSF, which largely funded her research. And that's really unusual for a PhD student to do that. Now, Meagan also wanted to get experience doing field work, so she spent 4 months working under really extremely challenging conditions in Cameroon. And she also had to make really important contributions to collecting data and samples. I'm really appreciative. She also had the foresight, or the drive, I guess, initiative to look at the impact of pathogens on microbiome diversity. So once again, she had the initiative. She said, I'm just going to take on a whole new subject. I'm going to learn about parasitology. And now she has some really interesting results due to that. So Meagan, I will let you take it from here.
Thank you very much for that very kind introduction, and thank you everyone for coming here today. I'm very excited to share my dissertation results with you. So the work I'm about to present today is largely based on the first 2 items that you see on this list, although I just want to note that I've had the fortunate experience of being able to be involved on many other different projects in my time here. So for the outline for today's talk, I'm going to introduce a little bit about what the gut microbiome is, why we're working in sub-Saharan Africa. I'm going to talk about my hypotheses that are specific to my research. I'm probably going to progressively push this dais forward and bring it back a few times. I might use the mobile. And then there's going to be two separate parts that I'm going to focus on, and those are the two papers that I highlighted in the prior slide. The first is on the population structure of human gut bacteria in a diverse cohort from rural Tanzania and Botswana. And the second part focuses on some recent work in Cameroon looking at gut microbiome composition and gastrointestinal parasite infection. And I'm just going to do some quick conclusions from those. So I started doing microbiome research because I found it to be this fascinating nexus between human health and physiology and the environment. And thus it's an ideal place to study human microbial coevolution and adaptation. Infectious diseases have had a substantial impact on human evolutionary history, and neglected tropical diseases, one kind of infectious disease category, are a massive public health burden for non-industrialized countries. They've also been understudied and underfunded. Before the Industrial Revolution and the advent of hygienic developments, the antibiotics, infectious diseases were the biggest killer of people in the world. And for the first 90,000 years of our existence as a species, we've been hunter-gatherers. So it's only relatively recently that we switched lifestyles and sort of changed the disease landscape of the diseases that we were exposed to. In the last 10,000 years, we have begun to live in cities, practice farming and raising livestock, and that's changed the type and frequency of the pathogens that we get. So the hygiene hypothesis has broadly said that our increasing industrialization and reduction in infectious disease burden has been concurrent with the rise of chronic illness. These include allergic, metabolic, and autoimmune disorders. And this kind of idea that industrialization may be changing our disease landscape has been broadened by a more recent modification called the old friends hypothesis, which says that maybe all these enteric parasites are not necessarily completely pathogenic and deleterious. They might actually have some benefits, or we at least might be adapted to their presence. And the role of the human gut microbiome in parasitic illness has been largely unexplored. So humans are colonized by complex ecosystems of microorganisms called the microbiome. Human microbiomes have site-specific composition and function, which means that the gut bacteria that is on your skin, or the gut microbiome that's on your skin, is going to be compositionally and functionally different from the microbiota of your gut. The ratio of bacterial to human cells is around 1 to 1, and the ratio of human to microbiome genes to host genes is around 100 to 1, so they're doing a lot of work for us. When I say the gut microbiome, I also don't just mean bacteria, I also include fungi, archaea, eukaryotes, and viruses. And when people say the gut microbiome, they're usually referring to the distal colonic bacteria, which we can most easily sample through non-invasive stool sampling. We know from prior research that the gut microbiome is influenced by myriad factors, including diet, sex, age, geography, industrialization, immune response, genetics, and disease. But which ones play the strongest role in rural, diverse populations remains an open question. Comparatively less is known about if and to the extent to which protists, helminths, and other infectious pathogens can have a role or even possibly modulate the gut microbiota, and what this means for human health. Given that Africa is the origin of modern humans, and Africans harbor more substantial linguistic, cultural, phenotypic, genetic, and environmental diversity, they might also possess some unique adaptations in their gut microbiomes. So here I'm showing some of the distribution of locally adapted traits in Africa, and some of these have been involved in dietary adaptations. For instance, amylase and the ability to digest lactose sugar from dairy are both adaptations that have been found in Africans, as well as disease resistance, variation in height, high altitude adaptation, and skin pigmentation. However, despite this incredible amount of genetic diversity, African populations remain understudied in genomic studies. So here they make up in the orange sliver about 2% of recent GWAS catalog-based sequence studies that have been done. And this is largely reflected in the microbiome as well. So we don't really know a ton about African diversity. There's been gaps in our understanding as a result of this about the spectrum of variation within human gut microbiomes. So I'm going to use the term subsistence often, and I think that that's not a readily accessible term. So when I say subsistence, what I'm actually referring to is the lifestyles that people have to get and procure food and their activity patterns. So hunter-gatherers arose before 10,000 years ago. Any population that derives most of its food from foraged plants or hunted game animals are termed hunter-gatherers here. And I denote that with this little bow and arrow up top on the right. In this work today, I'm going to be showing you 4 different hunting and gathering populations. One is the San from Botswana, the Hadza from Tanzania, and the Ndabaka and Bajeli, who are sometimes known as pygmies based on their short stature, from Cameroon. Pastoralism arose around 8,000 to 10,000 years ago in Africa. They have economies that are centered around cattle herding, and they have diets that are rich in meat, blood, and dairy. And here I have the little pastoralist icon denoted by a cow. We have Herero pastoralist from Botswana, Maasai pastoralist from Tanzania, and Ngororo, or just Fulani, Ngororo Fulani, or Fulani from Cameroon. And finally, we term populations whose diet and economy are centered on small-scale subsistence farming as agropastoralists. They raise cattle, have small livestock, they have diets with grain, maize, which is what I'm using to denote them up here, grain, vegetables, fruits, and tubers. And we have Bantu that are from Cameroon, other Bantu that are from Botswana, Sandawe, and Burundi agropastoralists from Tanzania. So Bantu is actually a language family. We use it here because there's lots of different tribes that have Bantu language designations, and we have the best statistical representation when we don't parse those up into tribal numbers of 5 or 6. So that's why we keep them here. They have the same shared subsistence practice as well. So I'm going to go on to part 1, so looking at population structure of human gut bacteria in a diverse cohort from rural Tanzania and Botswana. And in this section, I'm going to test the hypotheses that rural African gut microbiomes will be adapted to different diet, geography, host genetics, sex, and subsistence. And that subsistence groups will have microbial genes enriched in pathways associated with industrialization, for example, environmental toxins. So the Tishkoff lab went to Tanzania and Botswana in between 2011 and 2013. They collected a total of 126 samples. 112 of those were Africans, and we got 12 from US in collaboration with Rick Bushman's group. We have paired subsistence groups again across both of the countries, which means that for each one we have at least 1 hunting and gathering, 1 agro-pastoral, and 1 pastoralist group. And I've noted those populations again in this column here. Just a point to note, we were able to collect fecal and plasma samples for most of our individuals that we sampled within Botswana and Tanzania. And we also were able to use our comparative healthy omnivorous Philadelphian data as our sort of industrialized data set. And those individuals have self-identified European, American, or African American ancestry. And there's a bit of a lag on me forwarding the slides, so bear with me. All right. So in the field, we would ask participants to give us a stool sample when they were able. We would take an aliquot of that and flash freeze it with nitrogen, then we would store in a -80 freezer until it got shipped on dry ice back to the United States. Once we were in the United States, we were able to extract the fecal DNA in a wet lab, and then we moved on to doing the amplicon-based sequencing. So at this point up here, we're extracting fecal DNA. This actual tube of fecal DNA contains the DNA from all the microbes of the gut microbiome, including some host DNA, which we're not actually trying to sequence in here. In this next step, when we move on to amplicon sequencing, we intentionally target the 16S ribosomal RNA of bacteria. And this gene codes for a structural ribosomal protein. It's a component that's found in nearly all bacteria and some archaea. And it has been annotated with these conserved regions in green and variable regions in gray. So what we do is we design a little nucleotide sequence called a primer that allows us to use this conserved sequence to capture the variable sequence. And the variable sequence will inform us about what kinds of bacteria or archaea are there. Following this, we add molecular barcodes onto these little snippets that we created, and then we're able to, after sequencing, this is a very simplified version of this, but then after sequencing, we're able to ID all of our different strands of DNA to know which individual they came from. And then what we do after that is we would compare the sequences we get to ribosomal databases. And those ribosomal databases would tell us what types of bacteria and archaea we have. And then we can use that data to ask questions like what organisms are present and what is their relative abundance and imputed gene function. So, preliminary results. When we look at the family level average OTU proportions across populations, we have two major taxa that stand out. The first is Protoplasma, which is the turquoise color found abundantly in the Africans in the first 7 columns. And the Bacteroideaceae, which is the next one, is this olive color that's highest in the US samples. Protoplasma is associated with diets that are high in fiber and plants, as well as simple sugars, and this is in line with what we would expect from prior research. US and African populations have similar levels of Rubel, which is a highly abundant gut bacteria that's also associated with diets high in simple sugars and carbohydrates. But Bacteroideaceae, which is what the US have a lot of, is associated with diets high in proteins and animal fat. So when we order the populations by proportion of the Protoplasma bacteria, the Hadza have the greatest enrichment of Protoplasma among the Africans here, and the Bantu have the least. So next we wanted to explore the within community diversity of samples. We used a diversity metric called the Shannon diversity. This takes into account the bacterial relative abundance. And if you consider in picture A, these would be, consider this the microbes from the guts of individual A and individual B. We would be able to see just based on the different number of types of bacteria that individual B has more diversity than individual A. We call this within individual diversity. So this is the measure of within individual diversity across all these different groupings. So the first thing that we noted is the US has lower diversity compared to the African samples. And as a whole, that's an agreement with previous cross-population studies that consistently find that industrial populations have decreased bacterial diversity as compared to non-industrialized populations. We also know that the Botswanans are less diverse than the Tanzanians. And when we break out the diversity within the African populations, we find that the Herero have the lowest mean Shannon diversity, and Sandawe over here have the highest. This is kind of surprising. We would have expected from prior research that Hadza gut microbiomes would have shown the highest diversity. We think that Hadza, as hunter-gatherers who still actively practice hunting and gathering, egalitarian food sharing, they're eating a lot of the same things, that may cause potentially some convergence of the way the microbiome looks. There can also be, due to high relatedness, the transmission of heritable bacteria taxa that make the gut microbiome look more similar. And also, as another idea, the Sandawe over here who have this high diversity are a former hunting and gathering group that was recently settled in the last 100 years. And they sometimes fall back to the bush to supplement their diets with foraged plant animals. So in that case, you're seeing what could potentially be a sort of dietary fluctuation, which could increase the diversity of that population since they don't have a steady state diet. If we break out the diversity within the African populations, down here, we're now going to do the bacterial abundance. And so this is essentially saying that if we took all of these bacteria and we plotted them on a phylogenetic tree, which is going to sort of parse out how similar these microbes are, these bacteria are, based on their genetics, their genes, it's going to say that ones in groups that are closer together are more similar, and then you could say this one and this blue one are much further apart, so they're much more divergent. This is giving you a different measure of bacteria. This is actually going to tell you what bacterial abundance is between individuals. So you're looking at between people instead of within. And what we found here was that on this y-axis, weighted unit frac distance, this is going to show you if you are entirely different in terms of the bacteria abundances that you have, or entirely the same. And we see that as a population, Africans and the US, first and foremost, they don't look the same, that the US tends to have more bacterial dissimilarity of abundances than the Africans do. And over here, we see that Tanzanians look more homogeneous in terms of their abundances of bacteria. In the population segment, we see that the Bantu look very different from the Hadza. The Hadza share similar abundances of bacteria, while the Botswana Bantu agropastoralists have much more variability in the abundance of bacteria in their guts. We use principal coordinate analysis to map the phylogenetic distances between individuals into two-dimensional space. Each individual is one of the points in this plot, and the shape correlates with their population. These are clustered together based on similarity in bacterial abundances. And the first principal component that we're seeing here is accounting for 45.2% of the variation in our data set. This reflects a gradient between 3 different taxa. And again, these might sound familiar: Prevotella, Bacteroides, and Rhinococcus. So what you're seeing is that as we move to the right and the abundance of Prevotella increases, these individuals that are on this arms, that would be this group up here, have higher Prevotella abundances. Conversely, as you move further to the left and look at these dots here, they're higher in Bacteroides abundance. And that corresponds with the individuals up here. And finally, Rhinococcus has a bit of an arc here. So the individuals that are highest in Rhinococcus are falling right along the bottom of this plot. So in general, we can say that most of the Africans in our sample have a high abundance of Prevotella and low abundances of Bacteroides. However, we had a subset of 12 US Bantu individuals, so the gray here is the US, and the little brown boxes are the Bantu, that were statistically indistinguishable in their phylogenetic distances from the US samples. We also, we tried to figure out what this was, being contributed to because in general this like flies against the conventional sort of prior reported knowledge that Bantu should be high in Prevotella and not Bacteroides. So we looked at possible correlation with age, sex, sampling site. We did qPCR for 9 different endemic parasites. We looked at proximity to a major urban city or a water source that was close to a major urban city. And none of this could explain why these 12 Bantu were correlated with the US. So this led us to explore the possibility that these Bantu individuals may be experiencing some measure of industrialization. If we visualize the net mean bacterial phylogenetic distance of each population from the US, taking over all principal components, we find that the Bantu individuals are most proximal to the US among the African populations. And here the Bantu have been split into 2 subsets, where Bantu 2, right there, are the individuals that were clustering with the US samples. We see that overall the Botswanans are the closest to the US between countries, and that Tanzanians are the most distant from the US. One thought that we had here is that the differences we see might be motivated in part again by the effects of industrialization. So we found that Botswana has a higher gross national income than Tanzania, as well as a larger percentage of its population that lives in urban areas. Botswana also has free clinical care, which could be increasing the utilization of clinical resources. And this made us think that even rural Botswana populations may be experiencing some degree of industrialization that's greater than Tanzanians. To investigate this, we looked at significantly varying taxa among our populations and then investigated microbiome function. So the analysis of composition of microbiomes, also known as the ANCOM method, was used to test for significantly differentially abundant bacteria across our groups. And I'm not showing all of the different bacteria here, I'm just showing some of them. We used the most resolved taxa that had to have at least 0.1% mean abundance and at least one population for this. And the first one I'm showing, here it goes. Sugarella was higher in Botswanans than Tanzanians, and prior studies have noted this bacteria is increased in urban as compared to rural Nigerians. We found that Treponema was higher in Tanzania and in hunter-gatherers. So Treponema has been associated with diets that are fiber-rich. Hunter-gatherers consume a really high volume of insoluble fibers. So here you're actually seeing what one of those tubers looks like when it comes out of the ground. What we'll do is we'll cut off a section of this, it'll look like this, and then we'll chew it, and they'll extract all the moisture and pulp out of that piece, that wadge of tuber, and then they will spit this part out. So how much of this insoluble fiber actually gets passed into the gut is an open question. However, it's thought that Treponema may facilitate some breakdown of this within the distal colon. Finally, moving on to our last 2, we found that Rhinococcus is highest in pastoralists. Rhinococcus is a common constituent of ruminant and human guts. And one thought about this is that maybe seeing it in the pastoralists at higher levels could be correlated with their close proximity to livestock. Finally, the very last one here, Erisipella tricaceae, has been very associated with both high-fat and low-fat diets. So its abundance in pastoralist beds is also an area that remains to be explored. We genotyped 97 of our African individuals on an alumina-5 omnisnipere, and we used this genetic data to test for correlations between host genetic distances and bacteria composition. In particular, we used linear regressions to look at relatedness between the bacterial phylogenetic distances, you can imagine that tree of distances again, with host genetic distances, which are again the relatedness between individuals. And then we looked at how all of those work with host geographic distances, which are longitude and latitude. And we found that for bacterial presence and absence, it was correlated with both host genetic relatedness and host geographic distance. And that bacterial abundance was significantly correlated with geographic distance, but not host genetic relatedness. And these results imply that overall, bacterial presence and absence is stratified both geographically and by host genetic similarity, while bacterial abundance is only significantly stratified geographically. So next we wanted to address gut microbiome function. We imputed metagenomes from bacterial taxa using a software called PyQuest, and then we annotated dysfunctional content using a database called KEG. We actually had, within Africa, 146 significantly different pathways between our different categories that we were looking at, and of interest to us were the xenobiotic pathways. So here on the left, I'm showing you bisphenol. Bisphenol is a pervasive pollutant in industrialized countries. You might have seen things on your water bottles that you buy saying this is BPA free. BPA is and BPS are endocrine disruptors, so there's been a big push to get them out of our plastics. The US has the highest pathway enrichment of bisphenol degradation. And within Africa, the Bantu have the highest enrichment of bisphenols, and the Bantu populations are the ones that are here in purple, so these three. Next, we looked at DDT over here on the right. Botswanans, again, they're over here in purple on this axis, have a higher enrichment of bacteria involved in DDT degradation. So DDT is a crystalline organochlorine. It was used as an insecticide in the 1970s, and then it was banned due to some environmental hazards it was posing on the local flora and fauna. Botswana, but not Tanzania, is one of 10 countries in the world that still intensively uses DDT. Primarily, they use it to spray the insides of the homes that they live in for vector control of the malaria-carrying mosquitoes that live all over their homes. And this suggests that selection could be acting on a metagenomic level, which could lead to an adaptive response to the presence of environmental xenobiotic compounds. So, to sum up, to address the first hypothesis, we found that several taxa differentiate populations, for example, high levels of Treponema and Hansa. We observed differences in bacterial presence and absence with geography and genetics, and differences in bacterial abundance related to geography only. For our second hypothesis, we found that some Bantu may be experiencing effects from industrialization, for example, Bacteroides abundances that are similar to the US. We found an enrichment of xenobiotic pathways in Botswanans may be correlated with increased industrialization and urbanization. So now I'm going to move to part 2, which is focused on gut microbiome composition in Cameroonians and how it is predictive of gastrointestinal parasites. And for this portion, I'm going to be testing the hypothesis that human gut microbiome composition will be correlated with pathogen presence and frequency, and with cytokine and biomarker responses within the host. So in this research, I focused primarily on soil-transmitted helminths and protozoa that occupy the distal colon, are physically proximal to the gut bacteria, and are associated with morbidity. And I'm showing you the 8 kinds of gut parasites that I focused on in the research, and their modes of transmission. There's many different types of gut parasites that can be transmitted to humans through the fecal-oral transmission route. Their transmission is significantly higher in communities that lack access to things such as piped water, soap, and clinical care. And I have some fun videos that I was able to shoot over the field on my phone. This is showing you using a camera phone, which is why I thought that study. I'm looking into a microscope, and this is a spongy Stercoralis egg. And the one that was over here that was moving all over, that's one that's no longer an egg, it's just is swimming around. So we do it from light microscopy. From a public health perspective, this is a really relevant issue. The World Health Organization has estimated that nearly a quarter of the world's population is infected with gut microbes, and this burden lies disproportionately within developing countries. Many of these gut parasites cause diarrheal illness, and it's the second biggest killer of kids under the age of 5 worldwide. Although gastroenteric parasites frequently co-occur, most prior studies on the relationship between the gut microbiome and parasites normally focus on the individual parasite exposures and do not consider them together in relationship to the gut microbiome. So Sarah mentioned earlier, in 2015, I had the privilege of going with members of the Tishkoff lab and working with a team of Cameroonians to collect ethnographic, anthropometric, and nutritional measures, as well as tissue samples from 575 Cameroonians. These include individuals from 2 hunting and gathering groups, the Baka and Bajeli, Bantu agro-pastoralists, and Fulani blood pastoralists. So again, the tissue samples included fecal and plasma samples, but we also expanded this data set to include measures of whole blood cell count, temperature. We also did thick and thin serum microscopy on blood and wet mount fecal microscopy. We paired this data with that of 27 urban omnivorous individuals from Philadelphia. And back in the States, we used a molecular technique called qPCR, which is very sensitive and specific. That allowed us to detect parasites from extracted fecal DNA. We used immunoassays to test for HIV, and then we were able to ascertain from this data that the Cameroonians had many different kinds and abundances of fecal and blood parasites, as well as HIV. However, when we consider their overall frequency of pathogen infection, we find that the highest rates of infection occur for soil-transmitted helminths, which are the ones that I've shown in purple. Considering the helminths further, we found that agropastoralists, shown in this portion, the red of these bars up here, and the hunter-gatherers, sorry, this is the other way around, agropastoralists yellow, hunter-gatherers red, that they had the most infections are through subsistence groups. The Fulani only had a couple of infections over here for Necator americanus. We noted that concurrent infection with more than one pathogen occurred around 39% of Cameroonians. And just as a quick note, this is called an upset plot, and I want to just mention that these are unique incidents. So how you would read this is, for this very first bar, we would see that right around 40 people were occupied by infected by just Trichuris. Whereas in the second bar, we're showing the proportions of different subsistence groups that were infected by both Ascaris and Trichuris. And you could have all 4 of these parasites at once, and we have around more than 20 people that did. So we next want to know if any of these co-occurring parasites were significantly co-occurring so we can probe them further. So when we tested this, which is being shown in the figure below, we noted that 21 pathogen pairs significantly co-occurred, and the ones in blue were positively co-occurring, meaning that they tended to go up with each other. So as you had an increase in one, but an increase in the other. And the most significantly positively co-occurring pairs were Ascaris lumbricoides, Necator americanus, Trichuris trigera, and Strongyloides stercoralis, which I'm going to refer to as the ants' helminths. We also found that HIV down here significantly negatively co-occurred with both Trichuris and Ascaris, meaning that as one of these would go up, the other one would go down. There it goes. So we did targeted amplicon sequencing from the slide I showed before, but this time instead of targeting one region called B1, B2, we looked at a different region called B4, and that also picks up some archaea. And when we did this, we showed that the US samples primary clustered together, again, not unexpectedly, it's the blue icon here. When colored by the relative abundances of Bacteroides over here in red, and Prevotella, which is blue, we can again see that gradient, where the US samples that are over here tend to be highest in Bacteroides. The Cameroonian samples, which are mostly in this group here, tend to be Prevotella high, although there is a little bit of variation in between them. We then also see the variation we saw in the microbiome could be attributable to the effects of having parasites. So in A, I'm showing the total bacteria diversity measured against total ants parasite count. So in this example, each parasite count is a presence-absence count of unique instances of a parasite. So each dot here represents an individual, This line is the regression line with a 95% confidence ellipsis around it. How you read this would be for 0, these are all individuals that have 0 parasites. As you move over, you would say that they have 1 within the ants group, 2 different ones, maybe Ascaris and Trichuris, 3 or 4. And what we saw when we did this analysis is that as you increase your diversity, you also get increased parasite count. Conversely, as you increase parasite count, you also get higher diversity. So we speculate that this higher diversity could be associated with disease morbidity. Factors such as parasite-induced peristalsis and epithelial shedding could produce an optimal environment for opportunistic microbial colonization. We then plotted all of our Cameron samples in another clustering analysis to see how their microbiomes look when coloring for parasite infection. So this is now a measure of overall composition rather than diversity that I'm showing over here. And again, the color corresponds to the population, and the shaded triangles indicate that they are positive for ants parasites. The unshaded triangles are negative. And what we found here is that this is a statistically significant clustering of samples, that there are differences in microbiome based on this clustering. But one of the things we were concerned about is that we knew that the Fulani, who are along these blue triangles here, were, as I showed before, very comparatively less parasitized than the rest of our individuals. So conversely, our hunter-gatherer groups had very high levels of healthiness. We had almost no healthy controls. So we found that the Bantu actually had a good number of both cases and controls, people who were and were not infected with ants. And I can tell you in a separate plot, we did the same visualization, we ran the same test, and we found that when looking just within the Bantu, we were still able to get a significant result. So you could separate a microbiome space significantly. They were clustering based on something that was going on within their gut microbiome composition. Okay. So we then wanted to know what gut microbiota best predict metadata variables of interest, and that includes ants, helminth infection. We used a kind of supervised learning method called the random forest classifier to identify taxa that were most important in identifying subsistence, which is on top, and ants infection, which is down here. So in the top one, A, I'm showing the 10 most important taxa in predicting Cameroonian subsistence from 16S rRNA data. They're shown by model importance, and then the box plot over here showing you the relative abundance of those 10 important taxa within the different subsistence groups. So you're able to predict subsistence with 82% accuracy, and this revealed that hunter-gatherers had higher abundances of Bacteroidalis, Prevotellus stercorea, Zucchini vibrio, face Lactobacteria, and Treponema. Pastoralists have high abundances of Adoribacter, Rickenolaceae, Bacteroides cacae, and Bacteroides orbatus. And agropastoralists have comparatively lower abundances for all of these taxa. We also considered the top 10 most important taxa in predicting ants infection in Bantu agropastoralists, again for the case control reason, and 16S rRNA data, which is in B, the lower one. And again, Bantu agropastoralists were the only group that we do this with. So what we found was that this model was able to predict ants infection with 81% accuracy. CF231, Bacteroidalis, Zucchini vibrio, Treponema, and Clostridium and Roseburia Thesis were all higher in ants positive individuals who were Bantu. So for sequencing methods, I've shown how 16S gene sequencing can inform about the types of bacteria and archaea within blood sample. However, in shotgun sequencing, you're able to sequence the entire microbial community, so instead of imputing gene content, you are able to actually sequence partial to full genomes. And instead of amplifying a particular DNA target, what we do here for shotgun sequencing is we essentially take a large DNA molecule, which is in our little tube of extracted fecal DNA, we fragment it, we sequence it, it again gets tagged with those little barcodes that allow us to ID which samples came from, which sequences came from which samples. Then we assemble these DNA sequences into overlapping groups to build an assembled sequence. And then this is what we would actually then either put into a reference genomic database, so we can actually see what kinds of viruses and phage, protists, helminths, fungi, and again, we also pick up bacteria and archaea. We could look at genomic databases that give us indicators of gene content. And that will allow us to identify relative frequencies and pathways of this full community. So that's one of the big advantages. We get a lot more data. So we shotgun sequenced a subset of 175 Cameroonians, and this included all subsistence groups, pathogen infections, and healthy individuals on HiSeq. We also integrated shotgun metagenomic data from 27 US samples from the Human Microbiome Project. We did this all using a crafting unique database. And when we look at the relative abundance of the most highly abundant taxa, they are all bacteria. HMP samples have high levels of Bacteroides in red, and we know that we see a lot more Prevotella in blue within our Cameroonian populations, especially our hunter-gatherers. The gray represents all the other less abundant taxa. So this is just showing the top 15 per population, which is why there's more than 15 total, because some of these populations have more abundant taxa than others. We know that the Baca had the greatest number of unclassified views at any level, and these results largely recapitulate the 16S gene sequencing, which is why you must be thinking, well, this is really expensive. What did you gain from doing this? We do get some very useful information from shotgun sequencing. So for instance, we get much more information on parasites in the gut microbiome. So when you use qPCR, you use a very specific primer and probe set, and I was going after a species level designation. So I wasn't broadly looking for almost all of these except for crypto at anything beyond a species level critter. So this information here, this is kind of complicated. This is showing up on this section. This is the cycle of threshold, or the threshold cycle. The lower this is, the earlier I got a qPCR signal, which means the more of the parasite I have in my sample. So the lower it is, the better it is on this axis. Over here, you're seeing the amount of total species level k-intercept we have from our Kaplan unique database. And in a perfect world, what you would see is that individuals who are highly positive for parasites, meaning that they would be somewhere down here on this axis, would also have a lot of reads because there would be a lot more parasite DNA. And for two of these, as Paris and Necator, you can kind of see that trend, but you see that it doesn't hold for some of our other parasites. And this kind of made us wonder for a while what was going on. So we actually realized once we checked out the genome sizes of some of these parasites, as Paris has 107.7 megabases, Necator is 213.9, and compare that to Triparis, which is around 4.4. Strongyloides 25.3, Crypto 7.2. So you can tell that these two have a lot more potential bias to just be covered, have their sequences covered in the sequencing run. So we think that's why we get that particular result. I also just wanted to note this dashed line here is showing the upper limit for our standards. So this is the point at which we could detect the absolute least amount of parasite in our sample, which is our limit of detection. This blue dot here represents the average cycle threshold for parasite samples where we had controls donated from the Penn Vet School. So we don't have it for all of them, but for Strongyloides, Cryptosporidium, and Giardia, we did have those controls to use, but they were not sequenced, shotgun sequenced. And anything in green is showing you samples that were also positive by light microscopy. So one of the other things we noticed is that for something like Entamoeba, while we did have many positive hits, we had a lot of hits that we did not have annotated as being qPCR positive, that were coming out as getting reads. And that sort of made us scratch our heads a little bit. So we looked at a couple of these, and then we decided to go back into the Kraken unique shotgun database and see if we could get any alignments to parasites beyond the species-specific ones. So now instead of saying Entamoeba histolytica, I'm going to say all Entamoeba. And when we did this, we found that there was co-infections with many other kinds of Entamoeba. And specifically, these are four that are known for colonized humans. And in recent work that's been done from another group that's working in Cameroon on humans and non-human primates, we know that these co-infections are not unusual, that you can have 2 or 3 different types within your gut. Not all of them may necessarily cause symptoms. And this was something that we then tested. We made a new RFC model to sort of say, okay, if we consider all these individuals to be positive, how well can we build a computer model that can say, given some gut microbiotic composition, you'll be positive for these Entamoeba. And even though Entamoeba has been considered a strong shaper of gut microbiome composition in prior papers, we found that it did not perform as strongly as ants did. It was in the low 70s, and again, our model for ants positivity was in the 80s. Another thing that using shotgun PK data allows us to do is to assign function to microbial gene content in ANTS positive versus ANTS negative individuals. So here the two rows respectively represent positive or negative ANTS positive individuals from the shotgun data on Cameroonians, and each column represents a functional pathway. So this is the name of that pathway, and this is the larger category that particular pathway falls into, so these are all metabolism pathways. And so we were trying to figure out what the meaning of was for what kind of information you could glean from this. And one of the things that stood out was that hunter-gatherers in particular, and ANTS-positive individuals, they were also hunter-gatherers, had this heightened amount of purine and pyrimidine metabolism. And we know that oxytrophic parasites, there are kinds of parasites that must salvage purine and pyrimidine nucleotides from the host to survive. So in our data set, purine salvaging parasites included the protozoans Giardia, Plasmodia, Intoniva, and Cryptosporidium, as well as the nematodes Strongyloides stercoralis. So it's possible, as one idea, that due to parasite scavenging on these nucleotides, the bacteria must produce more to ensure their own survival. And this may in part explain why we see this enrichment in pathways that synthesize these nucleotides. So then we measured 19 cytokines across 72 Cameroonians. So this was a subset of the shotgun sequence samples. We measured them and visualized them in separate correlation plots. Here you're seeing ANTS positivity, which I've highlighted as this vertical yellow bar. And we found that ANTS positivity was positively associated with Th1 cytokines IL-7 and IL-2, Th2 cytokines IL-5 and IL-13, Th17 cytokine IL-17A, and pro-inflammatory cytokines MIP1B and IL-1B. So out of all the cytokines that associated with ANTS helminth infection, we found that IL-5, which is this horizontal bar here, so here's the intersection of the ANTS binary and IL-5, explain the most microbiome variation in random forest regression than any of the cytokines. So IL-5 is a type 2 cytokine. Type 2 cytokines are associated with helminthiasis. It's essential in the development and recruitment of eosinophils to sites of infection. Stimulates the production of antimicrobial peptides and mucus in the intestinal epithelium during the period of infection. And we found that IL-5 levels increased with ANTS positivity and peaked around, they increased with ANTS positivity and they peaked at around an ANTS count of 2, so 2 parasites. Furthermore, we noted that several of the top 10 taxa that were produced in random forest models for IL-5 overlapped with those that we got from ANTS infection RFCs. And this indicates that cytokines and IL-5 in particular are associated with the gut microbiome and with helminth infection. So of our RFC results, we found that Bacteroidealis explained the most IL-5 RFC model variation in shotgun analysis, and the second most in the 16S gene analysis. Bacteroidealis also increased positively with IL-5. It's also the most explanatory taxon in ANTS infection in general. It's associated with inflammation broadly. But it also came up in our subsistence-based RFC, so it's a top result for just predicting what group we belong to. And therefore, because it's a bit confounded with subsistence, we can't rule out that we may be seeing this strong result due to the effects of subsistence groupings. When controlling for subsistence, the RFC results adjusted that for taxa, Pneumococcus bromii, CF231, Peptoclostridium acidophilum, and Candidatus azobacteroides pseudotrichonuclei may have roles in ANTS infection exclusive of subsistence. So Pneumococcus bromii has functions integrating foods that are high in resistant starch. It's been associated with fishing subsistence in coastal Cameroonians. And in our study, we found that individuals with this bacterium were less likely to be infected by ANTS with control-in for subsistence. Diets rich in starch-rich foods have been linked to reductions in gastrointestinal inflammation. Thus, it's possible that Pneumococcus bromii could have a protective effect against helminthic disease, or at least mitigate inflammation that is associated with helminthic disease. CF231 is a common occupant of ruminant guts that's occasionally been found in humans, but is not, to our knowledge, been associated with human fecal parasite infection. Peptoclostridium acidophilum is a versatile amino acid degrading anaerobe that also, to our knowledge, has not been associated with helminthiasis in prior research. However, Hadza hunter-gatherers were previously described as having an enrichment of peg orthologous genes involved in amino acid metabolism, and greater functional potential to metabolize proline, serine, glycine, and threonine. So it may have a similar function in the guts of infected Cameroonians. Candidus azobacteroides pseudotrichonymphae is a termite endosymbiont, unclassified taxa in the Rubel family, and non-pathogenic species in the genus Treponema, both of which are present in our Cameroon data, are considered common occupants of termite guts. Termites are a substantial component of hunter-gatherer diets and agropastoral diets in many parts of Cameroon. This is actually showing a baka boy within Cameroon going into a termite mound to get some food, the food being the termites. So Treponema, again, is a common constituent of healthy, normal, versus healthy non-human primate guts. And the termite consumption could be more common in individuals that are ANTS positive, but whether the presence of these taxa has any effect on the susceptibility or response to ANTS helminth infection is still an unexplored area. So to wrap up the answers to the hypotheses so far, we found strong correlations between microbiome composition, cytokines, and ANTS infection. We found that gut microbiome diversity increases as parasite infection increases. So to just wrap this up, here we worked out the microbiota of multiple groups to see how they change in association with different factors. This research suggests that people lose diversity in the gut microbiota as they become more industrialized. And loss of microbial diversity has been associated with numerous disorders in industrialized groups, including IBD and allergic disease. So understanding the vanishing world of traditional subsistence-associated microbes and how their biology works is highly informative for human physiology and adaptation to changing environments. My dissertation work has shown that geography is the most important determinant we can find, suggesting that colonization from the environment is extremely important. We also found that subsistence is important, which suggests that diet and acquisition of microbes from diet contributes to microbiome composition. Finally, helminth infection was associated with a response in the gut microbiota, and this provokes response in the host immune system. This response is not a simple tilting of Th1 and Th2 immune bounce, and our results suggest that Cameroonians were exposed to many infections, with multiple helminths being just one of them. Okay, the acknowledgements. There's a lot. I first and foremost like to thank my thesis committee, Sarah Tishkoff, Rick Bushman, Dan Biting, Theodore Scher. There's many members in here from the PennVet Path Bio group. Several of them either gave project advice, mentored me in parasitology, or gave me samples, which was very kind of you. So I'd like to thank Robert Greenberg, Chris Hunter, Sparky Love, Tom Nolan, and Boris Straitman. Penn Shop Microbiome Corps, everybody there has been hugely instrumental. I just especially wanted to highlight the ongoing help with Kyle Littinger, Jane Longkins, and Lisa Matei. And Tishkoff Lab and Bushman Lab, there are so many. I'm going to say thank you to all of you, because this is a very big list. But especially to recognize the people who have directly contributed to my project substantially. Matt Hansen, Eric Mbungai, William Knives, Marcia Holz-Bach-Beltran, John P. Dave, Michael Campbell, Simon Thompson, Jesse Connell, Arwa Boss, Lewis Taylor, Aubrey Bailey. You were all instrumental in making these projects happen. I really appreciate it. And I wanted to recognize my funding sources here, which through this I was able to do some very extensive and very big and very exciting experiments. So that was great. But we're not done. I'd like to also thank our University of Botswana colleagues, and also Thomas Mnambo in Tanzania. The Cameroon collaborators and field team, we all had a great time here despite some challenging field conditions at times, and it was a real joy to work with all of them. Most importantly, I'd like to thank all of our research participants in Tanzania, Botswana, and Cameroon. And we're not done. I have to say that it's been a privilege to be at Penn, just feeling like I've never been more intellectually stimulated and challenged to just take my science and my ideas to a whole new level, and that's largely attributable to the people here. Sarah took a chance on an anthro student who had a pretty far-fetched project from what her lab normally does and backed me up with every paper I wrote and grant that I submitted and presentation that I did, and she's really been invested in seeing me succeed, and I deeply appreciate her mentorship. These are just we had a lot of fun times, so a lot of these are just remembering that for people in the audience. Rick has been so instrumental in meeting able to parse the microbiome data and really turning me into the scientist I am now. I have to share a quick story that think you might remember. When I was about 8 years old, my aunt and my cousin, who were in this audience, and me all went to go visit my aunt's friend Kathy at the Salk Institute. And it was probably my first exposure to a real lab. I was like fascinated by the centrifuge and what kid stuff. But the highlight of that tour was going to this brand new ESL3 lab that had glass walls and papers taped to the inside, and they were working on HIV in there. And I could see the hazmat suits where people would prep in the prep chamber go in. And Kathy was describing about how they were going to work on HIV and solve a ton of problems with HIV. I was just, my little face pressed against the glass. I was so transfixed by this thought of helping people with infectious disease through science. And I thought, I want to do that someday. I didn't know it at the time. I actually didn't know until about 3 years ago. But that was Rick's lab at the Salk Institute. And Rick, I did not stalk you here. It was just that it all worked out. But I can't help but feel like it really maybe meant to be. I'm going to try not to get too emotional on these next ones. My friends from the anthropology department, beyond have been a source of inspiration. They've helped me ground my work methodologically. They've cheered for me no matter how far away we all are from each other. I've been continually amazed and inspired by the people I've met in graduate school. We've had some really wonderful adventures together, and they've motivated me with their examples to be persistent, goal-oriented, and always open to new ideas. Some of these folks have known me for over 20 years. They've seen me hit academic milestones, as well as being buddies in fitness, Eagles, Super Bowl celebrations, D&D, and general silliness. They've reminded me that there is more to life than just work, and my life has been richer for it. I've also had the love of my family every step of the way, and I have a really big family, many of whom came all the way out here today from California, Colorado, Nevada, and Arizona to see me defend my dissertation. Some of the most instrumental people in my journey couldn't be here today, but I know that they would be so proud to see me achieve this milestone. And I want to thank all of them for knowing that I could do this, even when I doubted myself. I promise we're almost. Finally, I have been so lucky to have the love and support of my best friend Kara and my husband Andy. For a few years, we all lived together in Philly, and it was a transformative period for us as young adults and as friends. They made me laugh through every hardship, they kept me supplied with coffee on late night deadlines, and they encouraged my passion for research. So thank you so much for helping me get here, especially to these two. With all the love and support I have from you, I know I can do anything. I didn't cry. Live streaming this on a private YouTube feed, so I told them that they wanted to ask questions, we can see if we get time and maybe get to some point.
And in the meantime, anybody here is welcome to ask questions.
Wait, you didn't say anything about the distribution of age groups.
Yeah.
Or they all
All studies is clear.
Yeah.
So for Tanzania and Botswana, we only had participants that were 18 up. And then for the Cameroonians, we had ages, the range was 4 to 90. So that was a much larger data set. In general, in terms of like the distribution of kids, we had like a good chunk of kids, but I couldn't test a lot of them because in terms of like case controls, because almost all of them that we tested ended up having multiple parasites. Yeah, a lot of them were hunter-gatherer kids, and they just had really high frequencies of parasites.
Oh, congratulations on that outstanding picture, man. And let me start by saying I'm very jealous of that big fish that you caught in your So just stepping way back, I guess a sort of central question in all of this is how much have human groups been diverging over time, and how much of their microbiota been diverging with them? How much is it inherited by descent? How much is it swapping around between individuals? And to what degree can your work parse that out? You have a really outstanding large data set, at least some of it's attached to genetics. So where are we left on that point? Because you found that geography was the biggest determinant for both presence, absence, and abundance measures. But for presence absence, you saw that genetics was tightly associated. So how strong was that? And like, what do you think about inheritance by descent over the millennia versus totally swapping around to the environment, and the environment has distinctive microbes that are populating?
Yeah, that's a good question, right? Good one. It has a significant effect. I think to quantify it over the entire genome, to say out of like all of the potential environmental and other, heritable factors that you throw in there, I don't think it's a huge one, at least at this point in the way that we designed this study. There's been other work that's come out of Julia Goodrich and Rubel's lab looking at heritable taxa. And in general, they find that the most heritable association that can be reproduced independently by other labs is with the human LCT gene, which is involved in producing lactase, which breaks down milk sugar, and bifidobacteria, which is associated with dairy. So that's a pretty strong connection there. To go about addressing something like how we look for heritability in this data set, I think what we would need to do is go back to our records and actually start to look at, especially with the genetics, looking at how much people are related to each other in terms of like trios of parents and offspring. So if you want to look at transmission, those would be your groups to look at. I know that Sarah has some ongoing work with trios in different African countries, including Cameroon, to do similar things with heritability in those populations. There's a lot of work that's also been done within the non-human primate lineage that's shown the sorts of divergences between different contemporary primates. And there's also work that's been done looking at microbiome composition from ancient coprolites and then calculus and other samples from ancient humans. So you can kind of see that as we have industrialized, we have progressively lost a lot of our Protostalaceae, got more Bacteroideaceae, that stays consistent. You see that the treponines that I mentioned in the Hadza are also found within the non-human primates, especially chimps and bonobos and I think maybe gorillas too. So that could also potentially be associated with termite eating. But I think that that's like a big question that we still have left to answer in the field. And we're going to need, I think, very specifically targeted experiments to sort of suss out trios of relationships between people, somehow control for the confounding that they all live in close proximity to each other if you're parents and children, to tease out the environmental component, and then come up with better methods to do really integrated and, most importantly, I think, independently reproducible methods that other labs can then use to find what sorts of correlations there are. Did I get all the major points?
Yes.
So that's a fascinating question, and I'm wondering if you can do it like with twins who don't grow up in the same family.
Yeah.
So then a twin that grows up in Africa and a twin that grows up in the States.
So that's a great experiment to set up. It's really hard to find the numbers of people who have had these natural sorts of separations in their lives that are identical twins. There has been twin work done in the UK. It's actually a really big study that was done with Rubel's group. And she found her heritable taxa association with a family called Piston-Settle-NCA. And so they found that there's an association there. But again, what happens is you have different methods, sampling sizes, populations that are influenced by different environmental variables. And so other groups have tried to reproduce these results using genome-wide association data or other data sets, other cohorts of people, and other cohorts of twins. And they've been a little bit more variable, which is why the bifido only really come through as the only clear correlation with the human LCT gene at this point. Which isn't to say that heritability is not there, that you cannot have heritable taxa of the gut microbiome. Just looks like at this point, they're a small component of how you get your particular gut microbial community types and abundances.
And it was my question I wanted to ask. Thank you for your answer. Since I have, we're going to ask the question I wanted to ask. I've heard that the study of the microbiome is kind of the Wild West of science right now. So it's really exciting that you're involved. Do you have some projections for us as your audience to what do you think the future of that is? What direction are we
I think that is it depends on who you're asking. I think as a scientist, I would say that you're going to see a lot more integration of omics level data sets. So you're going to start to see not just the microbiome being characterized, but the metabolome of the host. You're going to see the metametabolome, what are the small products, the small molecules that are being made by the microbiota. I think there's some really interesting work actually looking at the epithelial barrier and looking at how different microbes sort of colonize and persist within that particular space during different kinds of disease and health states, which is really fascinating. One area I'm personally interested in is the enteric microbiota of the parasites themselves. How much do parasites contribute to post-colonization? Does it actually change any of the functions within the microbiome? That's sort of an open area as well. If you were to ask somebody in biotech, I think you're going to see a lot more push towards trying to deliver personalized medical treatments in terms of, bacteria or other microbes that you can introduce into your body that could help with everything from, say, metabolic disease to losing weight. We already have a lot of evidence that talks about the efficacy of fecal microbe transplants. We haven't fully worked out the science behind why it works so well. It's not one specific group of bacteria. So I think the more we get into this, the more we're going to find that it's a consortia of microorganisms living in a complex ecology with one another that's doing a lot of these things. And I think the biggest trick to getting people to sort of get past the Wild West is being able to, again, generate reproducible, robust research with the proper controls and methods that are very detailed, sharing code, just being very open and transparent in order to make sure that we're all working towards the most rigorous science that we can.
Thank you for your passion for science.
So I actually think that maybe we should stop there. I'm going to invite the committee members to stay, and for all the rest of you, if you can come join us at about roughly 4 o'clock in room 302, is that right, of CRB? So 3rd floor, when you get out of the elevator, take a left, it'll be like the first room there.
302, CRD, 4 p.m.
Yeah, so we get 4 p.m.